Namaste, very good evening to all. Welcome to Yoga Scholars PGI MER. Today we have a very distinguished and honorable guest with us, Dr. Gurudutt ji. Dr. Gurudutt ji is the former head division of stem cells and gene therapy research, DRDO Delhi. He received his first postdoctoral research as a research fellow in the Department of Biochemistry and Molecular Biology. Subsequently, he did a second postdoctoral research as a research fellow of NSBRI, NASA, USA. And he was also awarded third postdoctoral fellowship of the National Research Council, NRC, Canada. He is the founder of Stem Cell Research Program, which is highly relevant to defense applications for the first time in DRDO, as he is the founder of the Division of Stem Cell Research in Institute of Nuclear Medicine and Allied Sciences, DRDO, New Delhi. We welcome you, sir, and we feel very honored to have you. Now I request you to enlighten us with your topic. It's a very interesting topic for all of us. Please, sir, go ahead.
Okay, thank you very much for your invitation too. Good evening to everybody. I am Dr. Gurudutt. I belong to DRDO. I'm a senior scientist, and I work in a lab known as Institute of Nuclear Medicine and Allied Sciences, located in New Delhi, and it is one of the laboratories of the Defense Research and Development Organization. It's a great pleasure here to be here on this online platform hosted by the Professor Akshay Anand of the PGI Chandigarh. At the very outset, I'm very thankful to him for his invitation to share my stem cell research experiences on this yoga platform. It is very interesting. The integrative medicine and yoga have become extremely important in these days, and even DRDO has taken up the yoga, integrative medicine, and meditation in a big scale under the umbrella of physiology. Well, coming to my topic today, I wish to apprise you how hypoxia that is being acquired inside the tissues and the body while doing pranayama and stem cells and rejuvenation aspects under the systemic level. Actually, my area of research is stem cell as applicable to defense, and in fact the yoga and meditation or etc are actually very new. But however, if I dig down, it has a direct connection with yoga, hypoxia, meditation, and rejuvenation. Well, we have been working on stem cells by stem cells to increase their application potentials by priming mechanism, by gene transfection mechanism, increasing the activity associated with certain functional proteins like BCL2, PU1, CD34, CXCR4, stem cell factor receptor, Ostrix, etc., both in mesenchymal stem cell and hematopoietic stem cells that are located in one of the depots of stem cells in the human body, that is bone marrow. We used too many techniques like modeling, cloning, side-directed mitogenesis, transfection, etc., to get the enhanced activity or functional phenotypes. Because everywhere there is in the body, stem cell are the microenvironment which is otherwise where they are located or where there is an injury, for example, even artificial creation of a stress site. They have all the things have certain limitations. Many limitations associated due to different stages of cell cycle, different availability of the different molecules on the site. So there is a suboptimal response associated with stem cells. So we have been working in this stem cell modification to get higher response potentials in the stem cells as applicable to defense injuries and their management. In this, we have also worked, for example, I will show you here, targeted mobilization of stem cells from blood to bone marrow in the transfusion through encapsulation of lipid or alginate based vehicles. This is I'm now coming to mobilization, okay, of stem cells from after transfusion from peripheral blood to the targeted site, that is bone marrow. Mobilization. This is a reverse mobilization. Usually in the clinics, The mobilization is done to take the stem cells from one depot, for example, bone marrow, to blood, and then take out and then transfer to another person. However, this is my area in general. I also work on mesenchymal stem cells located in the bone marrow here, for example, to augment the osteogenic or bone regeneration fracture repair capacities. The so therefore the stem cell is very important to take up the repair processes of the injuries, etc., provided you put them in high doses at the injury site. Now, stem cells naturally have a regenerative potentials in the human body, but their job is basically to carry out routinely occurring wear and tear, small magnitude repair mechanisms, injuries, etc. So therefore, they are being, by one or the other signaling mechanisms that's likely to occur inside the body will keep mobilizing from bone marrow or from any other stem cell depots. Many tissues have their own individual stem cells. Depot, then they migrate to the, circulation. They reach the, tissue which has been either damaged in a small magnitude, or the cells which are the tissue which is undergoing a senescence, apoptosis, or a stress, many factors. So they try to reach there and try to regenerate and rejuvenate the tissue. But I will come. On the request by Professor Anand, I dig down the stem cell aspects, including our work, and found the direct connection with stem cell mobilization in pranayama or yoga, under the influence of pranayama and yoga. That is hypoxia and mechanical stress causing the effects of the next PPTs. Okay. Okay, now see the pranayama, probably everyone already by this time must have got acquainted with being in the course of yoga, many tools, techniques, and stages. But one of them is pranayama, which is a deep
Hello. Sorry, I feel we are facing some technical issue. Hello. 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 Sir, can you hear me? Sir, you have to unmute your audio. You have to unmute your audio.
Okay.
Okay. Now, sir, you can share.
Audible?
Yes, sir. Now you can share your screen.
Am I audible?
Yes, sir. Yes, sir. You are audible.
Okay. So
please share your screen, sir.
The overall benefits of the practiced yoga, which consists of deep breath exercises and bodily movements, would result into back pain, asthma, cure of or reduction in the magnitude of back pain, asthma, depression, anxiety, etc., which all likely to happen because of the increased expression of certain cellular, biochemical pathways, proteins, catalytic enzymes, etc., which are shown on the right side, like increased erythropoietin, vascular endothelial growth factors, which results into blood vessel formations, hemoglobin synthesis, and etc., etc. Deep breathing exercises modulates, oxidative stress, which you must have already known. While it decreases the cortisols, and it increases the antioxidant melatonin and even glutathione levels. Coming to the oxidative stress, basically is the accumulation of reactive oxygen species, while there is a increased oxidative phosphorylation and energy metabolism. The increased oxidative damage, oxidative stress, that is reactive oxygen species accumulation, that is known as oxidative stress, would cause both the, usually people recognize it as it's going to harm the body or harm the tissues, but at the same time one should realize that the reactive oxygen species also the signal transducers, and it is because of that in the low intensity concentration it causes many of the signaling pathways to likely to to induce the signaling pathways and elicit most of the cellular responses, which are mostly beneficial. While the cortisol, which is a hormone, yoga and breathing, causes the decreases the cortisol, which causes the immunosuppression, hyperglycemia, and reduces the bone formation. The cortisol also reduces the neutrophil activity, etc. Therefore, the cortisol decrease is the mostly beneficial one associated with these yoga practices. While at the same time, there is also a coupled mechanism of increase in the antioxidant molecules like melatonin and glutathione in the cells, so as to balance the concentration of reactive oxygen species to reduce the oxidative stress and see that there is a balance is maintained, and then elicit a very responsible, very critical fate responses in the cells. Okay, existing tissue metabolism, it's likely to restore the homeostasis of the disturbed metabolic imbalances, and the cellular processor responses also contribute to the new tissue formation and regeneration by remodeling, leading to the replacement of cells undergoing wear and tear damage, apoptosis, senescence, active dormant stem cells. Also, it's attributed to regenerative potential stem cell trafficking, that's mobilization, as I was referring in the slide number 2. For stem cell from the stem cell depots like bone marrow, many tissue depots to circulation, and injured tissues are tissues which are undergoing wear and tear damage, apoptosis, senescence, and active to activate the dormant stem cells of the dormant tissues. Initiated by biochemical signals arise due to hypoxia and mechanical stimulation. This hypoxia, which what I refer to now, is it's actually one of the end result of the deep breathing exercises. Hypoxia is created because in deep breathing exercise you take the oxygen and you hold the oxygen supply for a while. It depends from 2 to 4 minutes from individual and practice to practice, and it depends on the ability of an individual who is practicing yoga. At the same time, the mechanical stimulation are associated with stretching and other mechanical or physical activities associated with yoga. So these things, the hypoxia and the mechanical stimulation, are responsible for most of the biochemical induction of biochemical signal and pathways, resulting into newer cellular processes and responses, which gives you beneficial and healthy effects. Therefore, at the end of the day and end of the course, at the end of the continued practice of yoga, you feel release of stress and good, are relaxed and feeling. You also feel rejuvenation. Regeneration has been occurred as a long-term effect. Okay. Now this since you take the in the breath hold in a pranayama, you hold the breaths for about 2 to 3 minutes, and when you repeatedly do it, it creates a intermittent hypoxia. That means there is a depriving the tissues and the cells with oxygen, and you suddenly supplies the oxygen. This creates the gradient of generation of oxidative stress, which goes down and which comes up, which goes down and which comes up to stimulate these cells. This hypoxia is one kind of a threshold stimulus for the cells, I will tell you. and secondly, the gradient, induced oxidative stress by supplying oxygen in excess after a gap of 2 to 3 minutes would also result into a kind of signaling threshold in the cells to elicit biochemical pathways. So this decrease and increase are sensed by certain molecular sensors and metabolic sensors in the cells, and downstream pathways are going to be initiated. Now, I think everyone knows hypoxia means it's starving the tissues without oxygen for a couple of minutes and then again supplying them and all. This usually goes in everybody's practice. Okay, once you do this, you feel you have been already feel good and relaxed and rejuvenated, everything. So why this rejuvenated process quickly? I mean, it quickly starts after doing this intermittent creation of intermittent hypoxia by breath holding pranayama exercises. Why you feel suddenly after some time, after sustained performing the pranayama, is that the erythropoietin, which carries oxygen to the various tissues, are in increased weight happens. There is a erythropoietin which is responsible to generate RBCs in higher number, or to generate erythropoietin of a higher affinity to the oxygen, and synthesis of it in higher amount takes place in the bone marrow. For example, I'm showing here the hypoxia is here. When you create an hypoxia, there is a factor known as von Lindau protein, which is a 24 kilodalton, which inhibits the hypoxia inducing factor A, which is a transcription factor. It binds to this and it inhibits. When there is an hypoxia, there is a liberation of this inhibition with hypoxia inhibitor inducing factor, which is a transcription factor of that 93 kilodaltons. What does it do is, it has since it's a transcription factor, it elicits several downstream genes, but at the same time, it also a transcription factor for the synthesis of erythropoietin in larger amounts. So I told you a synthesis of EPO, that is known as erythropoietin, and RBCs, which leads to enhanced oxygen transport and restores the oxygen homeostasis. Then you feel relaxed because it happens in a higher amount suddenly. So you give a stress and you get a relaxed end result. So that is the basic molecular mechanism which actually operates this way. And there are other hypoxia inducing factors known as hypoxia HIF 2 and 3, which are also activated together, but their activity in terms of synthesizing erythropoietin varies from a type of splice variants generated and different subunits generated and their assemblies together, but there is no guarantee as such. Some may work, sometimes they may not work, but hypoxia inducing factor 1 H alpha is the predominant one. And now HIF1 also, as a transcription factor, I said it also controls several downstream genes. It induces several downstream genes associated with certain cellular critical functions, but here I will refer only to including proteases. I will tell you what these proteases do later. So then when you supply through enhanced erythropoietin and hemoglobin and then RBC synthesis, there is a more supply of energy, more metabolic reactions taking place in the cellular system in a mitochondrial respiration, and then energy supply takes place in higher amount. You feel relaxed and you feel good. Okay, now this is regarding one of the processes why you feel rejuvenated after this exercise. Okay, now going ahead, as I said in the beginning, stem cells actually also regenerate the tissues which are weaker in activity, which are undergoing tear and wear mechanism, or which is under stress or whatever it is. So the stem cells come into play to generate many such tissues. But however, stem cell of that type, though place of origin depots may be different, but by and large, the phenotypically and genetically, the stem cell is by and large is the same, which actually does this job, other than the one which is known as hematopoietic stem cell, which carries basically the synthesis of defense cells and the RBCs, etc. This second category of cells which synthesize most of the tissues, given the microenvironmental optimum environment, they would do this job. So these are known as mesenchymal stem cells, basically are located in the bone marrow and also in the other depots of the body. I will show you all those such depots, and these, by the virtue of their certain properties intrinsic to stem cells and extrinsic to them, they regulate the formation of various tissues which are have a different origin of the germline origin. For example, mesoderm, ectoderm, and endoderm one. So intrinsic properties like pluripotency, multipotency, and tissue plasticity is very important with these cells, because they not only just differentiate into one type, they also differentiate into many different types of tissues or cells which are of a different embryonic origin. So therefore, in this context, they have also been called as pluripotency in the limited sense, though they are multipotent. But since different germinal origin tissues and cells can be generated by using these cells, they are also known as, in the limited context, pluripotency. But however, their response to generate these cells also depends on the surrounding environment. Therefore, they are known as niche elements, and there are certain factors which can prime them in the culture or in the surroundings, and also biomaterial based polymers and support devices. If you provide, they would respond even better. And therefore, these stimulating factors, as far as this stem cell is concerned, to regenerate the wear and tear and senescent or apoptotic tissues is extrinsic, depends on the extrinsic factors, also on the intrinsic factors. Okay, now coming to hypoxia. You see, I said, plasticity and the pluripotency is important. While it is true, but they also have to be, come out of their, depot and then, travel to the site of where they are required. Now, for example, let us take one depot, that is bone marrow. Let us also take the other depots, but the mechanism, biomolecular mechanisms remains the same irrespective of the depots. If you see here, hematopoietic niche, and this is a mesenchymal niche in the bone marrow. One is from the iliac bone and other is tibia bone. Let us only see the hematopoietic niche. It's by and large the same everywhere within the bone marrow. Hypoxia, that what you get or acquired during the yoga or intermittent yoga or pranayama, you see there is a creation of hypoxia, for example. These pluripotent or multipotent stem cells, which I have written a narrow arrow here, these are stromal cells which resides away from the blood vessels. Near the blood vessels, there is a high oxidative stress, there is an oxygen supply. They don't usually stay near to the blood vessel, they stay away in the bone marrow. Similarly, hematopoietic cells, which are also a quiescent, having capacity for self-renewal without undergoing any differentiation, also stay away from the blood vessel and near to in the region of perivascular region. So see, here is the oxygen concentration, which gives in a big decreased arrow. Here is also oxygen in big decreased arrow. So oxygen here it is more, oxygen it is more, while it is decreased towards the perivascular region from the blood vessel. So most of the stem cells reside, which are having a potential for self-renewal and to potential for, make the regenerate multiple tissues. They are actually stemness maintained cells which remain away from the oxidative stress or oxygen. And the hypoxia actually takes care of these properties in the stem cells. Therefore, hypoxia is very important, whether it is hematopoietic stem cell or mesenchymal stem cell, that is also known as stromal cells. So hypoxic region is about 0.5% as far as stromal cell is concerned, and maximum oxygen tension which goes up to 6%, which is near to the close to the blood vessel. So the hypoxia that is created, for example, if you keep creating hypoxia through intermittent yoga, pranayama, breathe exercise, whatever is being created, it will it will help to maintain these stem cells in a more quiescent stemness manner. At the same time, when you release the intermittent hypoxia by inhaling a larger amount of oxygen, there is a creation of oxygen tension, and oxygen supply keeps going in the bone marrow. Oxygen supply keeps going up to 6 or even more than that. So during this course of time, what happens is most of the stromal cells get stimulated and they convert into progenitor formation. And the progenitor formation lose their ability to stay in bone marrow. They enter into the circulation, and once they enter into the circulation, they look for their signals to home or to stay away, to stay in a particular injury site or a tissue where they have to regenerate the tissues. While hypoxia cells, I'm telling you one of the best mechanisms. Hypoxia, though maintains quiescent stem cells, it doesn't induce any mobilization. But what in that also, these cells which were under hypoxic region will also get mobilized through a different mechanism. I will tell you in the next slide. These are other depots where stem cells you can see in the placenta, in the embryo, in the bone marrow, in the retina, in the hair buds, adipose tissues, and there are various such depots occurring in the human body. Various everywhere, lung, neuronal. I would say liver. I would say kidney, cardiac. Everywhere there are stem cell depots. Though they are phenotypically similar to fibroblasts or the mesenchymal stem cells, their responsive abilities to the microenvironmental cues are very different because they have been grown, inbred in their respective tissue niches. So by and large, it's the same. But these depots-derived stem cells do not have much capacity to go to other place and regenerate the tissues, while mesenchymal stems present in the bone marrow have this capacity. I was telling earlier, the oxygen supply will, increases the oxidative stress and brings those, mobilizes the stem cells into the circulation. While the hypoxia, stem cells which are residing in the hypoxic region, though they do not lose the quiescent self-renewal and pluripotency, but still they get mobilized. I will tell you the mechanism. Okay. See, rejuvenation due to the intermittent hypoxia, that is reoxygen gradient, increases the reactive oxygen species due to anaerobic respiration in stem cells. Mobilization of regenerative potential stem cells from bone marrow and depot to the circulation happens, as I said. Intermittent hypoxia and flux of the reactive oxygen species increased during the reoxygen after intermittent hypoxia together are stimulus threshold to metabolically activate the pluripotent stem cells and egress from bone marrow, which is a main depot. And these circulating stem cells act as a patrolling cells in the circulation, and wherever they are needed, they will home to that molecular environment and try to convert themselves into the host tissue and build their own cells and see that key that is being generated. Intermittent hypoxia affects to preserve the quiescence and stemness in the cells. Repeated shocks cause telomerase activity induction almost in all the cells. This is one of the important aspects of how intermittent hypoxia induction and abolishing. That means, breath holding, deprivation of oxygen and supplying oxygen in cells. It does the activation of telomerases, which induces which will be induced in all the cells, which increases their lifespan. This is one of the reasons why the yoga, pranayama will all cause the increased lifespan, etc., etc. But molecular level, this is what is going to happen. So the next one, what I was telling to you is why these hypoxia, reason based stem cells, though they are quiescent and self-renewal, how do they migrate? They're not supposed to migrate. Hypoxia doesn't induce the migration, but indirectly it is one of the main ingredients how it induces their migration to the better stem cells. For example, you see hypoxia inducing factor, which is a transcription factor, gets upregulated during intermittent hypoxia. What it does, in one sense, I said that it will increase the erythropoietin synthesis and increases the RBC generation, which are capable and which are fresh and carry oxygen with high affinity and supply to the cells required. Therefore, you feel fresh and etc. But the same HIF1 downstream, it has some more target genes, which includes, for example, SDF1, a chemokine, which is also known as CXCL12, proteases, matrix metalloproteases, cathepsin D, dipeptyl peptidase 4. These are also the downstream targets of HIF1 alpha. So once these proteases are synthesized, what does it do? These proteases go to the these hypoxic region resided stem cells and cleave their stable interacted receptor ligand which are stably holding these most primitive cells. This protease mediated cleavage actually does they loosens the connections. They tend to come out of their space from there, which is catalyzed. Their movement is catalyzed by SDF1. This is not a protease. HIF1 is also a transcription factor for SDF1, stromal derived growth factor 1, or stromal derived factor 1. So it has a binding capacity with all the stem cells, including hematopoietic, including mesenchymal, which bears the CXCR4 receptor, which is a G protein coupled receptor. The moment when SDF1 binds, it makes the cells to undergo certain morphological changes and changes in the integrin expression, and therefore it keeps moving away from the hypoxic region to the vascular region, and then makes the transendothelial migration. So therefore, increased secretion concentration, and then responding to that through CXCR4 receptor, and then going and getting into the circulation. So this is the second mechanism where HIF1 or hypoxia induces the migration of better stem cells rather than stem cells which are stimulated and got into a stage of progenitus. So these are stemness maintained stem cells which are indirectly stimulated by hypoxia and gets into the circulation, which are more potential for better regeneration. So this is second mechanism, and they go into the circulation and they actually reach the wherever they are required. There are other pharmacological agents like EMD31, CSF, etc., including light, intense exercise, massage with a pressure of 1 to 5 Pascal strength will also induce, tissue residing stem cells, not the bone marrow one. That's how the massage also will contribute, light also will contribute, intense exercises which involves the bending and stretching will also contribute to mobilization of stem cells from the various other depots wherever there is a direct impact of the stress or the massage. The hypoxia-induced reactive oxygen species in the intermittent stages will also have certain adverse effects, the toxic pathological effects, but at the same time, as a downstream mechanism to hypoxia and hypoxia-induced factor, there are some antioxidant synthesis also takes place. For example, glutathione, even melatonin for that matter, they also takes care of the excess reactive oxygen species, thereby maintaining the balance between the hypoxia and the excess concentration of reactive oxygen species. Hope everyone knows what is reactive oxygen species. These are oxygen molecules generated during the excess respiration in the mitochondria of the cells, but carrying uncharged non-coupled singlet electron, so that they become reactive to any molecule that they come across ionically. So therefore they are known as reactive oxygen species, but at the same time they are also molecular signal inducers, not that they are pathological agents. So therefore balance is required, but at the same time hypoxia induced RSO species also signal inducers. But I'm telling in connection with stem cells which is resulting out of hypoxia. Generated especially through intermittent hypoxia and pranayama. You see the one of the experiment which demonstrates that especially the stem cells which are maintained in the culture in the oxygen supply at the beginning, and then subsequently transformed into a hypoxic condition for 24 hours incubation in a hypoxic condition, and then see if their responses in the in vitro in a transmigration assay. So when you see this as a in vitro model study, the effect of hypoxia at one level, that what I spoke so far was the systemic level. At the experimental level, taking into consideration the excess concentration of SDF1 synthesis by hypoxia inducing factor, which is SDF1, which forms the gradient signal to move the stem cells from the perivascular region into the circulation. This is an in vitro experiment to see whether does it really the SDF1 is being given to the cells. Now SDF1 is another thing. Now the cells which have experienced hypoxia will also have a higher responsive potentials to SDF1 molecule. So for example, you see if you transfer migration assay, the cells which are maintained in normoxia will have a lower cell number migration from lower chamber to upper chamber, where SDF1 is maintained in a higher concentration. Whereas MSCs, mesenchymal stem cells, subjected to hypoxia for 24 hours with a concentration of 1% oxygen, their movements or their accumulation in the upper chamber are responding to SDF1 molecule gradient is more. For example, here this is for 4 hours, this is for 6 hours. There is almost more than 2.5 double fold increase in their movements and accumulation. That means Not only that, the molecule gets synthesized in higher concentration and pulls the stem cells. It is also the hypoxia which through unknown expressions on the cells will also have a receptive ability more and more for molecules. That's what hypoxia maintained mesenchymal stem cells, their ability, evaluation of their ability to the molecule in the cells. So it is a dual effect, both synthesis of molecules which attracts the cells, but on the other hand, cell which also gets activated by hypoxia to receive the signal. It's a synergistic effect of both. Now this the hypoxia will induce hypoxia induction factor 1 alpha, who's comparing to normalxia, for example. It is tremendous increase is taking place here. If you see here, similarly, if you see here, S M to control, which is in normalxia. So this is the how the hypoxia inducing factor gets the synthesis, induction of synthesis takes place. While SDF1 molecule, if you see here again, hypoxia, in case of hypoxia incubated cells, there is an increased SDF1 secretion also took place. While CXCR4, that is a receptor on the mesenchymal stem cells which responds to SDF1 binding. So also will get higher synthesis levels, so that there is a synergistic pull from SDF1, the mesenchymal stem cells. At the same time, CXCR that is present in the mesenchymal responds to SDF1 binding. So this has affinity to this, this has affinity to this. This has more binding ability in terms of number, this has more binding ability in terms of number. So there is a synergistic process takes place, and therefore there is a response of mobilization, whether it is hypoxia maintained stem nest cells or even the stimulated cells under, when the hypoxia is getting normalized, when there is a supply of oxygen. It is by and large the same. This is, this is our own work. Whatever I was speaking about so far is was at systemic level, systemic level, and carried out by both in vivo and in vitro experiment. But systemic level means in all the tissues it could happen. Hypoxia could generate. But in this case, this is our work is very interesting. Only one slide, 2 minutes or 1 minute. We made a targeted hypoxia creation in a bone marrow by pumping or targeting quercetin, which is an antioxidant molecule, and also a metal chelator. We made up nanoparticles with PLGA and coated with Poloxamer 407. The Poloxamer 407, we are the first in the world that it sequesters the platelet microparticles present in the circulation and makes the cloud, that is platelet cloud, platelet microparticular cloud. And this cloud exclusively goes to the bone marrow. It is, you see here, the biodistribution studies of PLGA, quercetin, and PLGA quercetin, PLGA, and outside layer Poloxamer 407 coated. It extremely goes to the bone marrow. So what we did is by selective targeting quercetin, which is an antioxidant and it is a metal chelator, and therefore, it creates an hypoxia kind of environment. So we wanted to see targeted creation of hypoxia in a bone marrow. Does it really induce any kind of mobilization of the stem cells? Now we did the reverse here. Instead of mobilize to check the mobility of the cells from bone marrow to the circulation, which actually is required and happens in hypoxia and the pranayama. But in this case, what we did was to check the synthesis of hypoxia inducing factor, which in turn synthesizes the stromal derived factor 1, SDF1, which then cells actually respond. So, HIF1, as I told you, in the case of hypoxia, its synthesis gets increased. Now we created here targeted hypoxia in the bone marrow, which is eventually we expect that there is a HIF1 induced SDF1 and SDF1 gets accumulated, and there is a question of mobilization of the existing stem cell there after not only binding with SDF1, and then since this SDF1 is a secretory SDF1, not the membrane cell bound SDF1. So they will bind this and the secretory SDF1 carries these cells to the bone marrow. But we did the reverse. But ultimate aim was here to induce a targeted hypoxia in a bone marrow and then see whether SDF1 gets secreted or not. But what marker did we use to measure SDF1? We also did the, ELISA assay to measure SDF1. But to show you, I only took the data of cell migration, CD34 cells which are in the peripheral circulation. When there is a SDF1 secreted in higher amount coming from bone marrow to the circulation, the CD34 cells which are present in the circulation gets attracted to this and enter more in number into the bone marrow. That means SDF1 is getting secreted by this targeted hypoxia creation. So this was the basic thing. Based on the previous knowledge and all, we did this work too. For example, here we please kindly see this picture. The SDF1 is gets synthesized by the stromal cells. These are particles. You see, these are SDF1 molecules, and actually they go and accumulate on the extracellular surface, sorry, the interior surface of the endothelial cell was of the vascular wall. And these stem cells which are circulating and the which is CD30, for example, which starts responding to this particle SDF1 and gets closer to the endothelial cells, gets attached and enters into the bone marrow. This is actually mobilization. This is a reverse of the mobilization. But my question was not that. My question here was to answer this SDF1, whether enters or not, when you target poloxamer 407 coated quercetin PLJ nanoparticle, which exclusively goes by 90% to bone marrow. It should induce HIF1 alpha. Subsequently, it should induce SDF1. Therefore, these CD34 cells have been responded to going more into the bone marrow. So that means targeted hypoxia creates the induction of SDF1. So this SDF1, what does it do is, does natural job. But at the same time, this SDF1, since it is a secretory molecule, binds to the mesenchymal stem cells present in the bone marrow, either in the hypoxic region or in the non-hypoxic region, and brings out into the circulation, resulting into the mobilization of the mesenchymal stem cells. So this is a targeted study to create an hypoxia, and we also achieved, and we are happy that the hypoxia can be targeted, and targetably it can be created through targeted means. Okay. Conclusions here are: hypoxia generate reactive oxygen species which behave as signal inducers, maintains and regulates the stem cell functional characters, induce large number of genes or secretory products or factors and chemokines, induces antioxidants. Similar effects are also observed in all the tissues. For a positive effects, hypoxia and balance of reactive oxygen species requires to be maintained. Well, these mesenchymal stem cells, in general, as well as in the yoga practitioners who aggressively practice yoga, physical exercises, and pranayama, these mesenchymal stem cells, when they keep continuously patrolling in the circulation, bring about certain immunomodulation characteristics. They have a properties of certain immune anti-inflammatory secreting cytokines, and which actually suppresses the unwanted inflammatory responses. And it is also very useful in applying for the external tissue regeneration, because you see in the wounds and the external topical applications, you can bring out its very positive beneficial effects by applying externally. These are collected through apheresis or any other techniques. And internal mobilization, as I said, can be achieved through hypoxia, intermittent hypoxia, and targeted hypoxia. And they also will not only go to the interior wounds or interior tissues which are damaging, but also can be taken out, collected in bulk, and then can also be applied externally to heal the external injuries like wounds, skin damages, cuts, whatever resulted, which are all resulted due to any other reasons. Basically, external injuries usually are caused by heavy impact, blasts, heavy impact agents, while internal injuries can be senescence, apoptosis, degeneration of tissues due to imbalance of the availability of the cytokines, growth factor, etc. Mobilized stem cells act as drug carriers to the site of application. This is very interesting. Mesenchymal stem cells, if you transfect with any drug carriers, genes, and then again infuse, they actually go looking to the site of injury, and then they locate and they release the drugs or genes over there. So additional ability or the additional advantage of MSCs is that they look for a a site where they have to lodge themselves. They know by through molecular interactions, and then they release the either their own immune suppressing factors or which are encapsulated in MSC, certain drugs or genes, etc. So these are advantages, and this is all how the hypoxia can be mobilize the stem cells, both which are stimulated. That is means the stemness to progenitor level as you enter, which is come close to the vascular region in the depots, and also mesenchymal stem cells which have maintained extreme stemness can also be mobilized not only through by through the indirect secondary means mechanisms like secretion of proteases and breaking down its ligand receptor interaction, which is stable, which holds them there in the bone marrow because of they have been dislodged through molecular interactions, and also additionally their movements towards the vascular will be catalyzed by SDF1, which is also secreted due to the hypoxia through HIF1, and see that they are entered into the bone marrow, into the circulation, reach the tissues which are to be regenerated. So the yoga, pranayama, hypoxia creation is a catalyst for this internal self elicited healing responses to feel good in the larger sense, in the larger time factor. So therefore yoga pranayama created hypoxia is so important. So at the end, wish you all to stay fit and fine, and I all thank you. I'm Dr. Gurudatta. Thank you very much.
Thank you, sir. Thank you for enlightening us about the stem cells and its effects on yogic practices. So we have a question from our viewer. If I may ask you, it's can yoga, pranayama, and meditation therapy stimulate stem cell trafficking from bone marrow, providing complete regeneration?
Yeah, absolutely.
Okay.
But at the same time, this is not the eyes. I took this depot, that is bone marrow, is an example. But stem cells residing in other depots, as I showed you, there are so many depots. The stem cells are there, which if you couple hypoxia, see molecular interactions, whether they can result anywhere due to hypoxia remains the same. This is one of the contributing factors how mobilization can be affected from any depot. But at the same time, if you see the physical exercises, the massages, you induce the stress on a specific area on the body. So if there is a depot around, massage is not done everywhere on the body. Massages are done through wherever there are muscles. Muscles have a lot of stem cells. Their muscles are also one of the most important stem cell depots. So massages having certain pressure, maybe 1 to 5 Pascal strength, that's usually naturally are usually done on the massage parlors or wherever, whatever. So will also induce the activation through these same molecular mechanisms, and also could induce their mobilization. But at the same time, this local activation will contribute to growth of those own depot tissues, if at all they're required. If they're mobilized and gone to an adjacent tissue, if at all there is a requirement, they still home there and start converting themselves molecularly, physiologically, phenotypically, and differentiate into their respective tissue types. Okay?
Yes, sir. Thank you, sir. Thank you for enlightening us, and thank you for taking some time out, because we know you are very busy these days. Thank you for your time. We are very happy to hear from you and to hear your deep knowledge about giving us the deep knowledge about the stem cells and its effects on yogic practices. Thank you for joining us, sir.
Okay, thank you very much to everybody. Thank you.
Thank you.
Okay, bye.